Hi, my name is Uday Sandeep and I worked on this phenotypes and gene analysis. Let us go through the project, how I worked and what are the insights I have made. Firstly, I imported all the required modules. Secondly, I performed this hashing with my name and it is giving larger negative integer. So I go with the hashlib library and it is giving me big integer. So I planned it to come between 0, 1 and 1 to 100. So I perform this code and I get 58. So I take 58 as a seed value for my random sampling. Here I yeah, reading CSV file of the gene embeddings, and here I am reading CSV file of phenotype embeddings, and here I randomly sampled with my seed value 58 here, and I saved this CSV files for the randomly sampled one for future reference. And here I checked for the I reloaded the sampled CSV file over here, and here we have to perform data cleaning, as whatever the embeddings they are present here, they are not in a numeric arrays, they are in the string format, so we have to convert it from string format to numeric arrays in order to perform analysis. Okay, so I made this here, and after that checked about the infos and all, and matching this gene and phenotype data frames for the further analysis, and it would be easy for us if there is gene and gene embedding and phenotype and pheno embedding. I renamed the columns also for good accessibility and all. And here we are computing cosine similarity between gene and phenotype embeddings. Cosine similarity is nothing but the angle between any two vectors or any 2 points. If there are any 2 points in the space, we have to draw a vector for both of them to the origin, and the angle between two of them is called cosine similarity in simple words. So we have to find the cosine similarity, and here are some examples. Uh, Next, we are plotting the cosine similarity, and here is the insight. We can see that most of the records are lying between 0.25 and 0.3 of the cosine similarities, so that we can say that there is an insight that many genes are contributing to many phenotypes. So next, we are visualizing gene phenotype pairs based on cosine similarity. So these are the highest cosine similarity genes and phenotypes. So there may be insight like this gene is a casual gene to this phenotype. There might be chances to conclude it more strongly. We have to go through all the data and find the cosine similarity for this gene to this phenotype wholly, then we have to strongly say that. But here we sampled 500 records only, so we can't say it more accurately. Okay, then performing supervised learning to draw some insights, and I got an accuracy of 53%. So I randomly assigned some data and labels to in order to train the model. I found this, it is 53% accurate, in which we can say this is giving 53% accuracy means uh, for a random alignment of data and random assignment of labels, model accuracy is 53%. So we can strongly say there might be some relation for gene embeddings and phenotype embeddings, as they might have to be developed by the description of both the gene and phenotypes, and moreover there are so many genes somewhere related to so many phenotypes. So that is an insight here. Um, and I am combining gene embedding and phenotype embedding into a single column for further analysis. And here I am applying PCA, dimensionality reduction, and here I am applying k-means clustering, which it is giving two clusters here. And there are some insights here also, and I applying so here I'm curious about how the random forest classifier will go with the data. that I prepared. So I checked this accuracy and accuracy related metrics here, and here I do this TSNE dimensionality reduction, and I plotted it, and here I did k-means clustering with TSNE reduction, and I am getting two clusters, and most likely in the TSNE component 1 of 0, the left side is one cluster and right side is one cluster. This is also an insight. So if we want to reduce the dimension by 1 itself, we can take this component 1 of TSNE, and next this is a measured DF, and here I plotted TSNE visualizations here, and next this is a 3D view. So things are overlapped here. If we see from this view, there will be a plane separating these two clusters. So next, deriving new vectors from already existing vectors here and plotted them, and here I made a derived vectors visualization in which it is showing there are three different clusters which we don't want. So we want two clusters only, but it is showing three clusters. So there is no need to derive new vectors. That is an insight from this. All vectors are playing good, and here is the heat map based upon the distances with distance matrix, and here are some phenotypes and their related genes with the smallest distance. So there are more possibility that this gene is very related to this phenotype. So but to conclude it more strongly, we have to go through the whole data, because this is a random sample of 500 records. um, and here I implemented k-means clustering and DB scan. So DB scan goes good with two clusters and k-means is giving three clusters. So DB scan is good here. Next, comparing, then I also do this agglomerative clustering also, and it is giving three clusters which we don't want. And here are some of the conclusions I draw from there. There are strong relations of embeddings of gene and phenotype, which we already discussed above. And here is another insight, like DB scan with PCA dimensional reduction is good, and k-means clustering with TSNE dimensional clustering is good for further analysis and predictions. And deriving new vectors is not required because they are giving three clusters. And we observe from the above output, the more similar is MCCD D1 gene and decanalocamitine phenotype based on the cosine similarity of embeddings, and suggested some of the most related genes to phenotypes based on the cosine similarity, and suggested some phenotypes and their most nearer or relative gene types. These can be three casual genes too. Like there can be a chances for casual genes too based upon the distances and all. So these are the conclusion I have made, and this is all my project. So that's it. Thank you.